Hi everyone, so in this video I will be going over what you need to include in your second lab report on enzymes. So you should have received feedback on your first lab report. So make sure that you read through the feedback and recognize where you may have come short and received a lower grade on the rubric. Okay, so remember each time you submit one of these lab reports, it includes the same rubric. So the same thing will be graded on in this upcoming lab report as well. So if you forgot to include a title or it was vague, make sure that you learn from that mistake and don't make the same mistake twice. Okay, so always it's a good idea to look through the rubric, grade your lab report before you actually submit it. Okay. And then just like with the first lab report, you're going to add your submission using the link. Ignore the date there. So this particular lab report is on our T5S5 enzyme catalyst life experiment. So this was in your notebook, so page or your workbook, page 181 through 188. And in this particular lab, you already were divided into an assigned experiment. So even though there were 4 variables, you only completed one of the 4 experiments. So half of you did the substrate concentration experiment and half of you did the enzyme concentration experiment. So even though you only completed one of the experiments, you're writing a report on all 4 of the experiments. So that includes the data that was collected by your fellow students on the other experiment, as well as data that we provided you on the pH and temperature experiments. Okay, so you're going to be incorporating all of those. Now, if you take a look at page 183, that's where your lab report information is in your workbook. So you'll follow along, and as you're typing up your report, make sure you have that open, as well as going back and reflecting on what you need to include in each section of your report by having that rubric available. Okay, as well as perhaps this video. So with this particular lab setup, we're going to take a look at this question. Okay, so I'm going to go ahead and bring up our PowerPoint. Okay, so we are going to be, the name of the lab that this is on is the Enzymes Catalysts of Life Lab Report. So let's take a look at the lab report. So this is the same setup as the last lab report. So each lab report needs to have a title, an abstract, an introduction, methods, results, discussion, conclusion, and a reference section, as well as a picture of yourself performing the experiment. Okay, so those are all required elements for your lab report, just like last time. So make sure that you include all of these elements and in the same order as listed here. Okay. All right, so let's start out with the first part, the title. So remember, the title contains the independent variables. So what I like to do when I construct a title for whatever I'm doing, I like to list what the independent variables were for this report, as well as the dependent variables, and then just put it together in one sentence. So the first one, the independent variables that we examined in this particular experiments were the changes in the pH. So we had different pH, changes in temperature, that was a separate experiment. Substrate concentration was a separate experiment, and enzyme concentration was a separate experiment. So all 4 of those are considered independent variables within our lab report. And then what was the dependent variable, the responding variable? Well, that was how the enzyme functioned, okay? And we looked at the amount of foam that was produced. So that's demonstrating that the enzyme actually did its job. It converted the hydrogen peroxide into the oxygen and the water. So when I create this title, I want to include both the independent variable and the dependent variable. So usually I just put it all in one sentence. So I might say something like the effect of changes in pH, temperature, substrate, and enzyme concentration on enzyme activity. Okay, it could be something as simple as that. Okay, but it should be about a sentence long and include both the independent and dependent variables. So that would be considered a straightforward, good title to go with. Another option, if you're looking for something even easier, is go back into your Moodle page, bring that back up, and you can just turn this question into a statement. So it says our question that we're asking is, how does pH, temperature, substrate concentration, and enzyme concentration, that's the independent variable, affect potato enzyme activity, the dependent variable? So you could just make that a statement, or you could just have it as a question if you want. That could be a suitable title as well. So you can, it doesn't need to take that much effort to create a good, suitable title. All right. Let's move on to the introduction, or excuse me, the abstract. So like I stated with the last lab report, I recommend doing the abstract last as kind of a summary of what the entire lab report consists of. So once you've done the intro and the methods and the results and the discussion sections, then think about what your abstract should be. So you need to include, what did we do? What was the purpose of all of these experiments? How we did it, okay? So including the idea that we took the hydrogen peroxide substrate and we used the enzyme extracted from potatoes. So that was the enzyme involved, the substrate attached to the active site of that enzyme, and then the enzyme sped up the reaction so that the substrate was able to convert into water and and oxygen gas, which we measured, and then just give a brief summary of what the overall results were when we changed the variables around. Okay, so about a paragraph for that. Moving into the introduction. So the introduction establishes the research problem. So what question are we trying to ask? So basically we're trying to figure out the factors that affect enzyme function in that potato enzyme. So there are 4 factors we looked at: the pH, the temperature, the substrate availability, and the enzyme availability. So that's our research problem. How does pH affect the potato enzyme's activity? How does changes in temperature affect the potato enzyme's activity? So we're trying to just establish the research problems to make sure you include the variables that we are looking at. Next, we need to establish the scientific concepts, our background information that we already knew going into this particular lab. So for those of you that maybe don't have the strongest understanding of what a substrate, a product, an active site, enzymes work, that kind of thing, then you will want to go back into Moodle and examine S5, S4. Okay, so this was our basic enzyme functioning assignment. So, you can go back, watch this video again on how enzymes work, and then, take a look at what we learned about in those pages of our workbook. You can also use the textbook, the Adigia textbook, to help you understand a little bit more about the function of the enzymes. Okay, so those will help you in writing the introduction. Okay, so let's go back to what we should include. So I expect, some background information about enzymes. So what I mean about that is, what is a substrate? What are the products? What is happening with the enzyme? What does the enzyme do to the substrate to create those products? So you'll need to explain how enzymes work. So I should see things like the active site defined and maybe activation energy, so how it lowers the activation energy. So things like that. So give me some information, background information about how enzymes work. And then that way we can introduce what the enzyme we're working with in this experiment, as well as what the substrate is, and then the products that are produced. So you should mention that we are using the hydrogen peroxide as the substrate. Hydrogen peroxide, its chemical formula is H2O2. So it has 2 hydrogens, 2 oxygens. And then the product that it ends up, the enzyme helps the reaction that takes place. The substrate H2O2 will then break it into a water molecule and oxygen. So H2O, water, plus oxygen, O2. And we actually measure the oxygen in the foam that we're measuring. So that's the product, okay? The water and the oxygen are the product from that substrate. The enzyme itself that we're using is potato enzyme, okay? So you might want to look up what the actual name of the enzyme is and include that in this section. And then, since we're not just looking at hydrogen peroxide converting into water and oxygen under normal circumstances, we're changing the environment that it's in. So we're actually thinking about, how do enzymes become denatured? Okay, so you might want to go look at some background information about what causes an enzyme to become denatured. What does denaturization mean? What's happening to the folding of the protein, because enzymes are proteins. So some background information on that is also needed, because enzymes, they have a particular optimal conditions that they like to be in. So if those optimal conditions are not met, then how does the enzyme function, or lack of functioning? Why does that occur? So that should be included in the introduction. And then at the end of your introduction, you're going to simply state your hypothesis and provide logical reasoning for why you came up with that hypothesis. So you'll want to think about how enzyme function will be affected by change in pH. How enzyme function will be affected by change in temperature. How enzyme function will be affected by change in the amount of this substrate. And same with the enzyme concentration. So you'll have 4 hypotheses, one for each of these different variables. And then, briefly explain why. So, why do you think maybe a higher pH versus a lower pH will affect enzyme function? What do you think about the temperature? Why do you think high temperatures or low temperatures will, produce more foam? Okay, so things like that. So make sure you're clear on your reasoning. After you've completed the intro, again, it's about a page, maybe even a page and a half to 2, then you'll move on into the methods section. So with the methods section, we're looking at page 81. So page 81, or excuse me, 181. 181 is the pre-lab. Okay, so you'll need to write out exactly how you prepared the fresh enzyme solution. So not what's written here on page 181, but what did you do? So did you actually use a russet potato? Where did you get the russet potato? What approximate size was it? Did you use cheesecloth or something else? What did you use? Did you use a blender? Did you, add, how much water did you add? Did you use tap water? Did you use distilled water? So all of those little, pieces of information should be included on how you created that or got that enzyme. Okay, and then on page 184 to 185, you're going to take a look at, the actual experiment that you were assigned. Okay, so were you assigned the enzyme, changes in enzyme concentration or substrate concentration? So when you're doing that, you'll notice in the lab manual, it's basically just numbered, set of procedures. You're not going to include a set of procedures. You're going to identify what you did. Okay, so this will be in paragraph format, and you're in past tense because you already completed the experiment. So for example, if you did on page 185, the substrate, the effects of substrate amounts on enzyme activity, then go through the procedure list and jot down what you actually used. Did you use large test tubes? Did you actually label the test tubes, or did you just put a piece of paper in front with the number? Did you how did you fill them? Did you use like a little funnel cup, or did you even use a test tube? All of those little things. You're just going to describe exactly what you did, not what's written down here. Okay? And then once you've written down what you did completely, then you're going to briefly describe how the other experiments were conducted. Okay? So you don't actually need to go through and give a paragraph for every experiment. But just say what's different. Okay? So we use the same type of enzyme, but what was different between your experiment and the temperature experiment. Okay, so in the temperature experiment, instead of changing the amount of the substrate, we kept the substrate the same, right? So it was the same amount of what was it? So it was, we added 1 milliliter of the hydrogen peroxide. So that was the same amount of substrate to the 4 test tubes. And then the same amount of enzyme was added. But the test tubes were then placed at different temperatures. Okay. So just one sentence describing the main difference between your experiment and that experiment. Your experiment and the other experiment and that experiment. Okay, so that's all you have to do for that. Since we are using information from the workbook, I do expect you to cite the workbook. If you had happened to have taken pictures of the experiment before you actually performed it, that would be a good place to put that picture, is in the methods section. If you have just the experimental setup before you actually added the enzyme and saw the foam go. Okay. So that's an option if you do have that picture available. You don't have to include that picture at that stage if you don't want to. Moving on into the results section. So with the results section, remember we're always going to open with effective overall findings. So it's not looking at specific sets of data, but did you notice that enzyme function was altered based on the 4 variables? That's all you need to, just describe the general trends that you saw. That's it. Okay, did enzyme function differ with each change in variable? So 1 or 2 sentences right there. And then we're going to go into the visuals. So with the visuals, we actually have a table that you're going to type up and convert into a pretty picture. So you'll basically just use the table from page 187, but modified. Okay, so fill out that table. You'll notice that you have to modify it because we didn't set things up exactly the same as an in-person class. So the first table is the pH. Well, we didn't have group 1 and group 2, right, for pH. So with the pH data, you're just going to go back into our Moodle page, and then here is that data. Where did I put it? Right there. Okay, so here's the results from the pH. So we have our 2 groups. Okay, so group 1 and group 5, you'll just write 0 for pH 2, 43 for pH 5, and so forth. So you'll just basically just fill in that data right here. Same with the temperature. So the temperature is right here. So fill what temperatures we use. So 2 degrees Celsius, 22, and then what we ended up with. Okay. For the other ones, so substrate amount and enzyme amount, you have some of that data already, right? So what you're going to want to do is go back into page 186 and fill in your data for your particular experiment where it goes. Okay, so did you do the effects of the substrate or the effects of the enzyme amount? So whatever data you wrote down there, then you're going to average it between your trials and add it to the correct column. Okay. Now for the last set of data, that's where you're going to go back into Moodle and take a look at what your fellow members of your group did. Okay, so half of you did the substrate, the other half did the enzymes. So, you know, look up what another student did and write their information down. Make sure you write their name down too. And then, you know, average their data out. And then you'll have that for your final section of your table. So once you have this entire table filled out, go ahead and make it a digital viewpoint. So you could use Excel or a table function in Word document, something like that. But make sure you change where the specifics for our particular situation. Okay, so that'll be one of our visuals. The other visuals that we're going to include are 4 graphs. So we'll have one graph using the data from the pH. Okay, so we'll graph this data. The average, or the pH, will be on the x-axis, and then the average millimeters of foam produced will be on the y-axis. Okay, then you'll do another graph. The other graph will be on the temperature column. And then we'll do one on the substrate amount, and then we'll do one on the enzyme amount. So you're going to have 4 graphs, one for each of the variables. Think about, is it going to be bar graphs or line graphs? You might have to go back to lab number 1, or excuse me, topic number 1 to kind of decide, is this continuous data, is it discontinuous? Okay, and figure that out. So you'll have one table, basically this in digital form, and 4 graphs, one for each variable. Okay. In addition to the table and the graphs, you're going to include paragraphs. So each paragraph, I would section it off. One paragraph for the pH experiment, one for the temperature, and so forth. So for each variable, you're going to, first of all, identify where that data came from. So if you use data from the instructor, then you're going to state that this was given to you by the instructor. If you got data from another student, for the other experiment, make sure you name the student. I am using, so-and-so's data who was in my forum group for this experiment because I didn't conduct it myself. Or you're going to say, I did this experiment, this is the data I saw. And then if it's your own experiment, you can describe anything that you noticed that was, going on throughout the experiment. Whether the foam was produced right away for one particular test tube, whereas the other one, it took a little bit more time, or one overflowed, there's so much foam, whereas one you barely saw any bubbles forming. So those little observations can also be included in that paragraph. You're going to describe the overall trends that you see. So, was the enzyme activity, did it increase with an increase in substrate concentration? Did it decrease? That kind of thing. So what were the overall trends that you saw? So you use your visual graph and kind of translate it into, what the reader should be seeing in that graph. It's increasing, it's decreasing, and so forth. If you happen to have taken a picture during the experiment, like, you see your little test tubes with the foam bubbling out, you could place that picture here in this section because that'll, kind of go in with the results. Okay. And then make sure that the table and the 4 graphs include figure reference numbers. So table number 1, graph number 1, graph number 2, graph number 3, graph number 4, so that you can refer to them in the text. Okay, so if you take a look, according to graph number 1, you see that the overall trend is increasing due to increase in pH or something like that. With the actual visuals, make sure that you include titles. And for the graphs, don't forget to label the axes. Okay. And then finally, we have our discussion section. So with the discussion section, we go back, look at our 4 hypotheses that we had in our introduction, and decide if our data that was collected supports those hypotheses or not. So you're going to formally state whether you accept or reject your hypothesis based on the data that you see. So make sure that you're doing that for each one of your hypotheses, and include the reference to the data for that support. Okay. You'll also want to back up the statements with reference to appropriate findings. So you'll want to explain, you're basically in your discussion, you're interpreting that data. So explain what's happening and why it's happening. So explain what the data means in reference to the scientific concepts you wrote about. So for example, let's say that, well, let me just take a look at, let's go back and see the data here for pH. Okay, so here we see pH 2, nothing. pH 5, yeah, it bubbled. pH 7, it bubbled a lot. pH 12, yeah, it bubbled. But where do we see the biggest trend? Okay, so we saw that pH 7 had the most foam produced. pH 2, which is it's a strong acid, produced none. So why do you think this happened? Okay, so why did we get the neutral pH showing that the highest amount of foam produced? So extrapolate, what happened there? So the enzyme must work pretty well under, normal pH or neutral pH. But when we increase the pH to a more alkaline, look at what happened to the foam. Okay, the enzyme didn't work as effectively because we didn't produce as much foam. Look at what happened when we reduced the pH. We lowered it to a weak acid. We still see some activity, right? But not as effective as pH 7. So think about why. And then why did we get no activity under pH 2? So that might be where we add that denaturization into the mix. So you're going to take the data, and you're going to extrapolate and interpret it and use those scientific concepts to explain it. Okay? So explain the data. Okay, why does it make sense? Okay, so why does it make sense that we saw 0 foam produced under pH 2 and we saw lots of foam produced under pH 7? So you might have to go into your Adigia textbook, or maybe even do some research on the enzyme itself. Okay, the potato enzyme, there's information on that particular enzyme. What is the optimal pH for that particular enzyme? You could look it up. Just look it up on Google, and then make sure you include the information, the citation for where you got it from. And that will help us explain why at pH 2 we got nothing. Okay, so you're basically taking the data that you ended up with, and you're explaining why, in a scientific format, why we see the things that we see. Okay. So why did things occur the way they did? And then in addition to basically explaining the results, we're going to effectively link our answers of research question to solutions, problems, information from the field, and other applications. So now that we have this variable, we know that potato enzyme is affected by temperature, pH, changes in enzyme concentration, changes in substrate concentration. How can we use this to explain real world situations? So think about, this is a cell biology course. What happens when your cells get too hot? What happens to your cells when they get too cold? What happens when your cells suddenly are exposed to a lower pH than normal? Okay, so we got to apply this to our overall understanding of the real world. So how does this relate to our understanding of enzyme function? How does it affect our cells? Okay, so our enzymes are inside our cells, right? So each one of our cells, our skin cells, what if you pour hydrochloric acid onto your skin? What happens? Okay, it burns, right? Why is it burning? Because it's denaturing the proteins. It's causing them to unfold. So, this is applied back to our own bodies, our own cells in our cell biology course. Okay. And then finally, the last part of our discussion, we basically look at any potential errors that occurred during the experiments and ways that we can counteract that for future experiments, as well as what could we do next. So think of any potential errors that may have occurred either by you or by your fellow students that could have impacted the results. So this could include things like, maybe somebody in the class did not use a russet potato. They used a red potato instead. I don't know if that would change the results, but maybe it could have. Some students may have used, grated the potato instead of blended it. Could that have affected the results? I don't know. Maybe somebody didn't have test tubes, so they used a different device. So there's all these things that could have potentially impacted the results. So we're trying to just kind of isolate a few of them and mention them as potential errors that may have occurred. Then think about, what can be done next. So now that we know that, going back to the results here of pH, pH 2, no for the potato enzyme. Well, we know that, at low pHs, potato enzyme denatures. So maybe if we were going to conduct an experiment again, maybe we'll narrow it down to maybe pH 6, 7, and 8 and see how that varies. So maybe we can isolate the optimal pH. Or maybe we could use a different enzyme and see how that affects the reaction of the hydrogen peroxide. So instead of potato juice enzyme, could we find another enzyme to use instead? So those are all potential experiments that we could do to further, enhance our learning understanding of this concept. And then the conclusion should be about a paragraph, which includes what you learned overall from the experiment. So what are your key takeaways that you learned? What surprised you? Was there a particular data set that, kind of threw you off? And wow, nothing happened for pH 2. A lot happened for pH 7. So what surprised you? What will you remember for your next lab activity? So what maybe over doing the experiment did you learn to pay more attention to for the next experiment? And then we have our references. So minimum of 2 references, 3 preferable to get the full credit. So 3 references. 1 would be your workbook. Of course, everybody will have the workbook. Probably the textbook, because you probably use that for your background information. And then any other source that may have helped you. So for example, if you looked up the optimal temperature for the potato enzyme, I don't think that's in the textbook or the workbook. So where did you get that information from? Make sure you include your in-text citations where appropriate. So anytime you're using information from a source, you should have an in-text citation at the end of the sentence. Everything needs to be in APA format. So use, there's a number of different websites that you can use to get a perfectly formatted reference. So for example, I think there's easybib.com or something like that, where you just choose APA format, not MLA, but APA. You enter all of the parts that they require, and then it spits out the proper format. So you could use something like that if you need to. Don't forget to proofread your work. Okay, so make sure that you read through it very, very carefully, and make sure that there's no spelling or grammatical errors. Just be aware that, sometimes things are spelled correctly, but it's the wrong word. So don't, mess that up. So you got to proofread everything. Make sure section headings are in order. Boom, boom, boom. You include those. It's easier for the reader to find each of the sections. It's organized that way. Don't forget to indent your paragraphs. I'm noticing a lot of students don't do that anymore. I don't know why. Proper English format, we got to use our indentation of our paragraphs to denote a start of a new paragraph. Okay, so don't forget to proofread your work. And then just like last time and the next time, you need to include a picture of yourself doing the actual experiment. So that will be, you can place it, in the appropriate part of the lab report if you want. If it's you with the experimental setup or you with the, showing the results, that's with the test tubes and the foam, but it needs to be a picture of a selfie with you actually with the experiment. Okay, so it needs to be include, the experiment in the background. So I know that you actually did the experiment and did not just copy data down from someplace. All right. If you have, so once you've done your entire lab report, you're ready to submit. Don't forget to go back and look at the rubric, grade yourself. Okay. It's an easy way for you to make sure that you don't miss, easy questions or easy points, because sometimes I do see students, they completely forget the conclusion, or they lump the conclusion in with the discussion. Well, discussion and conclusion are two separate things. They're not just one title and then a bunch of information. Okay, so do make sure that you read the rubric, grade yourself. Make sure you're giving yourself, 2 points for the conclusion because you convincingly describe what has been learned on the lab with elaboration. Sorry, it's kind of weird how it's formatted there. Okay, so don't forget to grade your paper before you submit it. If you have any questions, feel free to reach out. I am more than welcome to meet with you, look at a rough draft, things like that. So I'm here to help in any way that I can.